Hi, I'm Sarah, and today I'm going to be taking you through how to answer a 6-mark exam question for GCSE biology with AQA. For the particular question that we're going to look at, we need to be aware of the specification points for 4.3.2.1 and 4.3.2.2. For the first point, producing monoclonal antibodies, it says in the specification that students should be able to describe how monoclonal antibodies are produced. It then goes on to state how the monoclonal antibodies are produced, and there are a number of keywords there. Please note the keywords that are within the specification, because examiners will expect to see these keywords used within your answers to questions on this particular content. For 4.3.2.2, use of monoclonal antibodies, it states within the specification that students should be able to describe some of the ways in which monoclonal antibodies can be used. Underneath, it gives you some different examples, but right at the bottom, it states that students are not expected to recall any specific tests or treatments, but when given appropriate information, they should be able to explain how they work. Let's recap the process of producing monoclonal antibodies. Here's a diagram to remind you of the different steps. First of all, we're injecting the mouse with an antigen. The mouse will produce lymphocytes, and those lymphocytes will produce the monoclonal antibodies, which have a complementary shape and are specific to that antigen. We need to remove the lymphocytes from the mouse. Because the lymphocytes won't divide rapidly, we need to fuse them with a tumour cell, as tumour cells do divide rapidly. This produces a hybridoma cell. The hybridoma cells will divide rapidly and produce the monoclonal antibodies. The clones of the hybridoma cell will produce millions of the monoclonal antibodies. Now we've gone back over the steps, let's take a look at the exam question. I always suggest reading through an exam question twice, once to get an idea of the whole question, and the second time to highlight any of the keywords and make some notes. Before answering any question, you should look at the command word and any important words that are going to influence how you answer the question. The command word here is describe, and it's followed up with the word how. If we're describing how, then we're going to be listing the steps or the stages involved in a process. So we need to describe the steps for scientists to produce monoclonal antibodies. Then we're asked to describe how these antibodies can be used to prevent the virus infecting human cells. The question tells us that we need to use the information from the diagram above, and we need to use our own knowledge, and this is important. It's quite common for AQA exam questions to state or specify something that you should do when you're answering a question. In order to get all of the marks available, you must always incorporate what they're telling you to do in your answer. It's worth 6 marks, and with 6-mark questions, they're often levelled, levels 1 to 3. If you get a level 3, that's going to be 5 or 6 marks. For levelled questions, you need to make sure that you've got lots of correct information, you're using lots of scientific detail and terminology, and that it's laid out in a concise and logical manner. So, for example, stages in a process need to be laid out in the order that they would need to occur in. And for this, I would recommend that you note down your key terms and organise them into the right order before you start to write your actual answer into the answer section. For this particular question, I would recommend breaking it down into 2 parts. Part 1 is describing how we're going to produce the monoclonal antibodies for the virus. And this is the part where we need to use our own knowledge. Part 2 is describing how the monoclonal antibodies can be used to prevent the virus infecting human cells. This relates to the second part of the specification that we looked at, where it states that students do not need to know this information already, but will need to use a diagram or information given in a question to be able to provide the answer. Looking at this diagram, we can break down the information that we're given here. So it's telling us that there's a human cell and there's a virus. The virus has antigen on its surface, and the human cell has antigen binding sites on its surface. We can see in the second part of the diagram that the virus uses its antigen to bind to the human cell via the binding sites. In the third part of the diagram, this has allowed the virus to enter the human cell. It's allowed the viral DNA or genetic material to enter the nucleus of the human cell. Let's look at answering the first part of the question. We'll need to recall all of the different key terms that we're going to use to describe the process of making monoclonal antibodies. I'm just going to list down any of the keywords that I can think of, and I'm going to try and get them in the right sequence before I construct my actual answer. But what I can do afterwards is go back through and number them so that they can be put into the correct order just before I use them to construct my actual answer. Now I've got my keyword list. I'm going to check through the order, see if I need to move any around. As I'm happy with the order, I'm going to start to write my actual answer. First, scientists need to obtain the antigen from the virus. They will inject it into a mouse. The mouse's lymphocytes. Lymphocytes are a type of white blood cell. It's good to give a little bit of extra detail where you can, and using brackets for this is fine. The lymphocytes are going to make antibodies, and these antibodies are going to be specific to the viral antigen. They're specific because they have a complementary shape. We will remove the lymphocytes and fuse them with a tumour cell. This produces a hybridoma cell. Hybridoma cells can divide rapidly, and when they do, this will produce lots of clones of hybridoma cells, which can produce millions of monoclonal antibodies. Something that you might notice is that my answer isn't laid out in sentences and paragraphs. It's pretty much a bullet-pointed list, and this is fine to do. This just makes it a lot easier for an examiner to read through. In the past, spelling, punctuation, and grammar were taken into account when marking answers to questions like these, but this is no longer the case. It's more important that you use the correct key terms and write your answer in a logical order. Let's look at the second part of the question. We just need to use the diagram. We need to say what's going on in the diagram and see how the monoclonal antibodies that have been made can be used to prevent this particular virus being able to enter the human cells. From the diagram, we can see that the antigen on the virus needs to be able to connect to the virus antigen binding site on the human cell. Therefore, monoclonal antibodies can be used to bind to the antigen on the viruses and prevent them from binding to the human cell. First of all, the patient will need to be injected with monoclonal antibodies. Just going to clarify again that the monoclonal antibodies are specific to that particular virus's antigen, and that this means that the antibodies will bind to the antigen, and the antigen are on the surface of the virus. This is going to prevent the antigen from being able to bind to the virus antigen binding site on the human cells, and in turn, this will prevent the virus from entering the human cell in order to replicate. Here you can see how the answer would look with both parts combined. You can see that I have put all of the key terms that I've used in bold. And I definitely recommend reading back through your answer to check for any errors that you've made and to make sure that you've used all of the keywords that you need to. You can circle or underline any of the keywords that you've used already, because this can help you check that you've included everything. And it's also going to make them stand out to the examiner who's marking your question. Here's the indicative content on the left-hand side. This comes from the mark scheme. It includes all of the potential answers that students may include in their answer to this question. There may be other things that could be relevant as well, and examiners will use their own subject knowledge in determining if what you've written can also be included and help you to get a higher grade or a higher mark in a question. You can see that the majority of points in the indicative content that have been mentioned in the answer. If we refer back to the question, it said that we had to use the information from the diagram, which I've done in the second part of my answer. And I also had to use my own knowledge on how monoclonal antibodies were made, and this was done in the first part of my answer. I'm Sarah, the science lead for Propello, and I hope that you found these tips on answering 6-mark describe questions for AQA GCSE biology helpful.